It's my pleasure to introduce Jacques Dumais. So Jacques Dumais is a big traveler across space and across disciplines.
Across time, I'm getting older too.
And so he did his master degree in British Columbia in morphogenesis in algae, and then went for his PhD to Stanford, and so, started to work with Paul Green, who is a big name in biomechanics of plant morphogenesis. And then, unfortunately, Paul Green passed away, so Jacques has had to find another advisor and to do things a little bit by himself. So he came here after his PhD for a postdoc, and actually he just told me that he gave a talk in this room about the same topic. So now you can see that you will be able to see how the picture changed in I don't know, in 15 years. So then he went back to Harvard as an assistant professor, where he spent a few years, and then moved to Chile, where he is now. So please.
Thank you, Scott. Thank you, everybody. So I noticed you were making fun of my title. Is there something wrong with this deconstructing?
Nobody knows what it means.
Yeah, okay. Me neither.
Only the literary faculty knows.
Exactly. That was the intention. So I did indeed give a talk about tip growth with transparencies and virtual projector here 15 years ago. Not that it didn't have proper projector, but it was still fashionable to use a blackboard. But you see, I had to move on. But so before I get started on tip growth, I want to invite you all to Chile. So I've been working in Universidad Adolfo Ibanez for the last 5 years. That's the university overlooking the Bay of Valparaiso, and we're right there on the coast, more or less in the middle, stuck in the middle of the Atacama Desert and Patagonia. Still quite a ways, but so if you like
What's the
adventure,
scale top to bottom?
4,000 kilometers. It's a long way.
And left to right?
Very short, 100 kilometers. So we're going to be talking about tip growth, and actually that's how this country was produced. So
Is it an
you're invited. Huh?
Is it an island?
Wait, I have a lot of slides. Can we move on? Okay, so I did go back to on Growth Inform, which I had read at one time, not from cover to cover, but I did look at quite an extensive part of it. And those who've looked at it will know that there is a chapter on the forms of cells. And there are two remarks that Thompson makes that I'd like to underline, because they're actually quite appropriate for this talk. So he begins his chapter on growth and form, or on forms of cells, distinguishing between agglutination, again, another word that's, so which I guess we call accretion now, or crystal growth. Huh? And he distinguished that with intussusception. A nice word. So intussusception is growth from within. And he makes the clear distinction that it's what goes on in a crystal growth, or this is a virus, the assembly of a virus, is quite distinct from what you see at the level of a single cell. And we understand quite well self-assembly of viruses. Perhaps there are a few things that we need to discover still, but by and large we know how to, or we understand how viruses are built. The reason for that is it's a small scale. So this, you probably can't read that, but this is 100 Armstrong, this bar here. So we're still looking at individual proteins that are coming together and perhaps minimizing energy and making a pattern. When you look at a cell, it starts around 1 micron, if you will, up to, this is a large cell, this is 400 microns here, this size. You're, in fact, above that scale, the scale at which self-assembly works well. So we still don't have very good theories to explain what goes on within a living cell. How do you build shape when you don't have this option of just self-assembling proteins together, or atoms together? So that's a question we've started looking at a while back. The effects of size, you'll see, is quite important. Now, most of the chapter on the forms of cells is about surface tension, which turns out is not so applicable to cells or living cells. In some contexts, it works. If you want to explain the shape of the cells that I'm about to describe, surface tension is not quite the right theory. But it does make a comparison. This is the pendant drop here, where he looks at or compares the shape of these drops with the shape of the shell of some foraminifers, to be taken with a grain of salt. I'm not sure that's the right format. But then he goes on to say one more thing that I'd like to underline. He starts talking about a budding yeast. So that's the mother cell of a budding yeast. And whenever you drink beer, you can think about that. Every now and then, they will make a little bud. So they need to divide. So they will start making a bud. And what he says about this process of budding in yeast is this. When a bud appears, it does so because at a certain part of the cell surface, the surface tension has more or less suddenly diminished, and the area of that portion expands accordingly. It is plain that over its surface, the surface energies, whatever that means, are unequally distributed owing to some heterogeneity of the surface. So I'll come back to that. But what he's claiming is that there must be some sort of gradient on the surface here, surface energies or molecules or substances, that makes it bulge out. So that will be the, if you will, the Thomson 100 years ago scooped me on this topic. Okay, so we're going to look at cells that are growing by what is called tip growth. Here's 3 examples, which little movies here. I'll use this. I'll use this here. So Eclea is an oomycete. Don't need to know what an oomycete is, but it's a big lineage of organism. Lily is a plant, as you know. This is a bond too of a plant. And finally, Allomyces here, growing by tip growth, is a fungus. Yeah. So they all do more or less the same thing. I just want to emphasize, you're looking at essentially 3 major lineages in the tree of life. In fact, Allomyces is more closely related to us than to plants. So these cells have all somehow converged on the same way of building shape. So that's what we're trying to understand. So let's see, I will just give you, there's not so much biology in this talk, but I'll start you with 3 elements that are important to remember. So here's here's a tip growing cell, and there are 3 features you'll have to remember. First of all, these cells are polarized. So they will often have, that's what I'm drawing here, a gradient of some molecules, and often they will have cytoskeleton elements like actin or microtubules aligned within one direction. And that's how they actually polarize and therefore grow in one direction. It's been studied. I won't talk so much about polarization. In fact, we're going to start with the premise: the cell is polarized. And then the question is, how do you make the shapes? How do you maintain this nice finger-like structure? And the second process that you'll need to know is the form is achieved by molding the cell wall. Okay, so these cells, they're not animal cells. They have a plasma membrane, which I'm barely drawing here in black. And then on the outside, they have a wall, which could be made of pectins. Is that a question?
Yes.
Yeah.
For number 1, just to make sure I understand, you mean we just have to agree that there's some internal process that maintains some chemical gradient? That's what you mean? Right?
That's right. Or more generally, let's agree that there is a vector pointing this way. So it's polarized. So it's been studied. There are many papers on how cells get polarized, and you'll see how.
Oh, is it electrical, or is it a bunch of things, vesicles and whatnot, that accumulate?
There are ion gradients, so in fact there is flow of ions, but they think it's mostly molecules that are localized here in the membrane that maintain the polarity. So there is a gradient of molecules within the membrane of the cell here that is.
That's a gradient of molecule concentration,
Concentration
rather than
Electrical.
electric field.
There are currents, but it's not the basis of the polarity. It's a chemical gradient. Okay, so that's the question really. We know that the second process here, it has to grow, it has to remold, if you will, or deform this cell wall, which I was mentioning, could be made of pectin. We'll talk about a little bit about pectin today, but it could be cellulose in some cells, and it could be glucan in other cells, range of different molecules. And finally, that's why we're here. It's a mechanical process. There is an internal pressure. I might say trigger at some point. I mean by that is a pressure that's established within the cell by osmosis. So the cell has an osmotic potential that's lower than its environment. Water rushes in, and as it does so, will stretch the cell wall. And that's going to be critical for explaining how you maintain the shape. Okay, so those are the three elements that we have to consider. If you look at, the question, if you want to understand morphogenesis, really, you're trying to explain how the shape is evolving in time. So here is a lily pollen tube, the same cell at different time points. For lily, they grow quite fast. That's about maybe a minute or two. So you can actually see them zoom by when you look at them through the microscope. Fish and yeast, a bit smaller, so actually an idea of scale here. This is between 10 and 20 microns, depending on the size. Fish and yeast, same thing, so cells growing, and this one at the two ends, at least at some stage. So if you want to understand how they form, you'll have to map the cell wall. I just mentioned the cell wall is really what maintains the shape, or gives the shape.
Excuse me, what's the scale on the lower one?
This one is about 3 microns in width, something like that, 3, 4. So what we've been doing for many years is you don't see it here, but there are these cells are decorated with little beads that are marking the cell surface, and we track them in time. So it's like PID, if you will, for a living cell. So we're saying this is a composite of this, in fact, this cell, I believe, growing over time, and all of these little paths here represent beads that got stuck on the wall and got moved in time.
Do you keep adding some, or you start by
In
fixing
some
your-
experiments, in this one we added some because then they all get advected and you get a naked tip after. This one is one staining, so those are quantum dots that are added to the tip. Okay. So we have the first ingredient, or the access to this morphogenetic process, and we have a way of marking the cell surface. And then the problem becomes more physical or mathematical. And I would say to formalize it, if you will, this is so tip growth is really, or tip growth morphogenesis is a map. And we want to understand that map. So you can imagine, for example, you define this a vector here that defines the position of a wall element at a given time point, and then you want to see how it moves or is advected over time. And so you have this map here that will depend on the initial position of where it was and depend on time as well. So that's one way to do it. So if you were to use always a reference frame and these little vectors, it's actually not very convenient. In fact, it's not, depends on the reference frames you set, so it's not a universal way of describing what goes on. So what you can do instead is take derivatives. So if you take two derivatives for this vector here along the length, so a spatial derivative, you'll get the curvature. And that's something that we can work with. It's intrinsic to the geometry of these cells. You can also differentiate, if you will, the map in time and get the velocity. So this point is actually moving down the side. It's going away from the axis of growth. So that's a velocity. So if you have, instead of using these functions, which are really just measuring the movement in x and y, so two functions that are describing how it moves in 2D space, you will use the curvature of the cell and it's the velocity at which the wall elements are moving down the side. So we've measured this. This is what they look like. And you'll see many of these graphs, so let me just explain what's going on here. So most of these graphs will have, this axis will be the arc position. So with 0 here being if you will, the pole of the cell, and then going positive and negative on either side. So this first graph is the velocity, and you see it with the bead. So the velocity is 0 on the axis. A bead that happens to get stuck quite close to the tip will stay there for a long time so I have 0 velocity essentially. And by the time they reach the equator, then they are left back and the tip keeps moving forward at a given velocity. So that's, you reach asymptotic velocity, if you will. That's the velocity of the tip. And in this case, it's about 15 or so microns, 0.15 or 0.1 microns per second. And the geometry looks like this, more curvature at the tip, and then obviously as you go down to the side here, it's like a cylinder, the curvature drops to 0. Okay, so that's the first element of what we need to describe this morphogenesis of these cells. We have the description of geometry in time, or at least we assume that it's steady, so the geometry here, curvature, stays essentially the same. And we have a description of a velocity field on the surface of the cell. Now, to bring it closer to mechanics, because we would like to understand the mechanics of how they grow, we in fact take the classical derivative of these values. For example, the gradient of the velocity gives you the strain rate on the surface. So that's the meridional strain rate, so how fast wall elements are stretching along the meridian here. And you can also look at the circumferential strain rate, so how fast you're moving and deforming this way. And this is what you see. So extensive growth near the tip, so at 0 on the axis of the cell, you have extensive growth, and then it drops down quite quickly to 0, so there's no growth. By the time you reach the side here, there's no growth. But there are a few features that are important. So one of them is, so there's the sharp gradient, but also you see that over almost the entire range here, the transverse or circumferential strain rate is higher than the meridional strain rate. So the cell, if you will, and I'll show a picture in a minute, I'll show you a picture here. If you were to look at a wall element, a little circle at the tip, what it goes on to that surface element, it actually gets deformed over time, more transversely than axially. Okay, so that's the map, if you will, or the deformation profile on the cell surface. The other thing is, I'll come back to that. In many cells, we register a small contraction. All of this was positive stretching of the cell surface, but in some cells, more or less on this the side here of the cell, you get a little bit of meridional contraction. I'll come back to that. Okay, so we've got these two forms here are essentially the same thing. The color map here is just the areal expansion of the cell surface, and I'm just using circles here to visualize the anisotropy, which is the ratio, if you will, of these two strain rates. So we've got that, and again, two functions describing how this cell is deforming, and now we'd like to take it down to something more physical. I'll try to explain what is the basis of that gradient. First of all, why does it drop down so quickly? Why do we see this anisotropy between the transverse and axial direction? So one thing you can do with these cells, because they're actually quite thin, and that's something that, in fact, that Darcy Thompson does to some extent in his book, you can look at the force equilibrium in the cell wall. So you have, it's like a rubber balloon, if you will. You have a pressure inside the cell, you have a thin wall, and so the forces have to balance, and it's not growing fast enough to have any other fancy effects, so it has to be in equilibrium. So that's my way of drawing the Laplace law with all the vectors. So that's the equilibrium between the pressure, which is normal to a wall of the surface. It has to be in equilibrium with the tension that are in the two principal directions. Yeah? So that's one equation you can have, and then with an axisymmetric cell, you could also consider, you take a slice of the tip, for example, and you also have to balance forces. There's pressure acting on the circle that you just slice off, if you will, and it's resisted by tensions in the wall, and you solve these equations or these equilibrium equation, and that's what you get. So those are the typical equation for a thin shell, pressurized thin shell. So that's the stress you would expect in the meridional direction, which will have obviously to scale with the pressure, and then it's inversely proportional to the thickness of the shell, and that's the radius of curvature in the circumferential direction. And there's a similar equation for the stress in the transverse direction. Okay. So we've measured on the previous slide, the field of growth, the formation of the cell wall. we have equations to predict, based on the geometry and the pressure, what the stresses are. We're like to connect that. That's something we started some time ago. So we have, so this is the stresses. This is the profile. Let me mention a couple things. So this doesn't start at 0. So again, that's the meridional position. That's the stresses. That's my computer. That's, I just keep sending emails. So this is the meridional stress. So that along this axis here, it's essentially constant. Doesn't vary very much from the tip to the equator. But the circumferential stress does change substantially near the pole. It's equal. It has to be equal to the meridional stress. And by the time you reach the equator, so on the two sides here, this way, it's twice as much. Okay, so there is, over most of the surface here, the wall elements are, so these little wall elements are seeing more stretch or more stress in this direction than in this direction. Okay, so can we, this is the true set of measurements that we have now. The strain rates on one side and the force equilibrium of stresses on the other side. And already you can see that there's a bit of a paradox, because you're growing fast and near the pole, and this is where, at least on average, you have the least stress. Yeah, so that's obviously not a simple stress. I'm stressing it, I'm putting force, therefore I'm flowing. It has to be a bit more subtle than that.
This is data from the live cells?
Yeah, it looks too clean.
It looks very clean.
But it's data. So the other thing is the anisotropy. So I've mentioned there is an excess circumferential strain rate, right? And that's at least fits well with the fact that we have an excess stress also in the circumferential direction. Now, this little paradox between having maximal growth at the tip and in fact having least stress was already resolved by Thompson. He says, well, it is plain that over its surface, the surface energies are unequally distributed owing to some heterogeneity in the substances. So you can imagine it must be softer, right? If the wall is softer here, the cell is doing something, well, even a low stress there is enough to give you high expansion rate. So now the question is what that is. So we're deconstructing, so taking apart. So we're trying to take apart the system, which seemed too complicated at the start. And so that's something we came up with more recently, so it's obviously it's not a viscous process. It's not a simple viscous or elastic process because of, well, some of the things I showed you and the fact that the cell will have to control its growth and therefore cannot afford to simply let mechanics dictate everything that goes on its surface. So we came up with a simple, what is being called a chemorheological model to explain how the wall is controlled. And so this is I'm going to explain that in more detail, but this is the basic equation. We're saying that the strain rates that we're observing on the surface will be equal to some, this is a concentration times rate constant, so that's, you could normalize that by the concentration of elements in the wall, if you will. So that's something that the cell controls. The cell can control what are the chemicals in its cell wall, and therefore this is a term that we regard as a biological term, something that the cell regulates. And then this other term, W, is a strain energy function, and therefore the gradient of this gives me the dissipation of energy. So those are the two elements which I'd like to detail a bit more. So that first element, growth, is affected by a reacting molecule, and that's why we have this first term here at the beginning, the first term in the equation. And then once it's flowing, it will flow such that it maximizes the dissipation of elastic energy in the wall, and therefore that's why you get this gradient of the strain energy function. So here's how we see it. If you look at a small piece of cell wall and look at it, the typical organization, I'm depicting actin here, so this would be actin in blue, and those are calcium ions that are allowing pectins to interact together. So what we hypothesize and believe is true is these actin molecules, or not the actin, pectin, I should say. This is pectin. So these molecules or the bonds are strong enough to resist the pressure. So they're deformed elastically, but they're not flowing. So this is not, think of it as a set of springs, and so with these bonds strong enough to be able to resist the pressure inside the cell. Now, when the cell wants to grow, what it can do is, through this term here, secrete new material, and one of them could be secreting pectins that's not connected or cross-linked. And you can imagine that a pectin that wants to bind calcium, and it finds itself besides a very very stretched calcium ions here, or calcium bonds with the pectin, it could actually interact with these and relax locally the stress that you had here in the wall. So the cell will have control over how much it secretes of this pectin. It in fact the pectin is secreted in a form that's not reactive, and so it has enzymes also that the cell can secrete, like pectin metallesterase, that will prime, if you will, these molecules so that they can then react. So there are a few ways the cell can control what will be a scalar. That will be a concentration of reacting molecule. And once you break a bond, mechanics takes over. So you broke a bond, there was elastic energy, you might expect the mechanics, if you will, to make the most of it, to dissipate as much energy, elastic energy, as you can, given that you've relaxed one of the bonds. Okay, so that's really what's summarized in this equation here. And so we try to see to what extent it's predictive of what the expansion we observe. And
Excuse me, why do you say the statement it maximizes energy dissipation? Is that a conjecture or a theorem or a what?
Let me step back. This is, in plasticity, they often write plastic flow in this form. So that's called what is it called? Associated flow rule, a flow rule.
I understand the principle of achieving a minimum energy state. And what you're saying is you're trying to get there as quickly as possible when you maximize.
Yeah, so imagine a mesh that's being stretched twice as much transversely than actually, if I break a bond, how is it going to deform? Well, it will relax more transversely than actually because the stresses were higher. And the basis for that is you have an amount of energy that you can dissipate because now you have more degrees of freedom. Yeah. What you're going to do with it, you just find a way to maximize the little freedom you have to relax.
So it's a sort of plausible conjecture.
Yeah.
You maximize it.
Yeah, that's right. Yeah.
Okay.
Let's call it a conjecture. Okay, so with this conjecture, we cannot afford to have too many parameters. So we took the simplest possible strain energy function that we could come up with, with a few additions, let's say. So we'll assume now we're looking at this is the strain, elastic strain. We'll just assume the strains I didn't mention. The strains are actually quite small, even if there's quite a sizable pressure in these cells. The strain in most of these cells is about 5%. So it's not like they're deforming like a balloon that you're inflating. They have about a 5% strain on their surface. So we're assuming it's just the linear. So this would be a compliance matrix, if you will. That's the stress. So the linear combination of the different stresses. But we'll add to that a few more things. We'll see that the properties of the material are plane isotropic, and that's because of this, right? The fibers that we have, pectins in this case, are all in the plane. They get deformed in the plane, and therefore they all align in that plane. So obviously perhaps you can have isotropic properties in the plane, but it ought to be different from what goes on across the thickness because you don't have a fully random network there. So we've added this plane isotropic properties, and then the elastic deformation, we say it's incompressible. Obviously the cell is adding mass because it's growing, but we just say that when you deform elastically, it's also incompressible. So you'll recognize the typical energy density function here with only two parameters. So there's mu here and nu, which are the only two parameters, and really the only one that will affect the result is nu here. I can detail that more later. So what I'm showing here is actually level curves for this energy function. So I'm looking at the stress space, and these are, each of these curves here represent strain energy, a constant strain energy level. If you want to maximize the dissipation of this strain energy, well, you'll want to take the gradient of that space, and you'll be physically what it looks like, or geometrically what it looks like is you're taking the little arrows that are normal to that surface. So you're going to try, for a given stress state here, you're going to try to flow in this direction to relax that stress. And so we've actually measured, or projected, if you will, the measurements that we had made in this space, and this is actually the data here. It's not a model anymore. I'm outlining here in blue the region of stress that are possible or that are observed for these cells. And what you're seeing here is the actual measurement. Let me step back and show you you can use these here and look at the anisotropy and define a ratio that gives you a cosine or it gives you an angle, and you can just project that here. So for a number of positions on the surface of the cell, we looked at the ratio of the two strain rates that we observed in the experiments. I put them here as a vector, and you'll see that by and large, they are all orthogonal to these curves here. So it's not perfect everywhere. There's a little deviation in some cases, but by and large, if you're given that these angles could be anywhere within this quadrant here, there's actually quite a bit of degrees of freedom, they follow quite closely these level curves. Okay, so to sum up, I had these two functions, which were the strain rates that we had measured, and with this model, if you tell me the geometry, in fact, and you give me 1 degree of freedom, which is this parameter nu, I can draw this strain energy landscape, and I can predict how the cell should deform. So give me a shape, give me one number, which is 0.6, more or less, 0.6, 0.7. That's the nu here that we observe that fits best our data. And I'll give you how the cell deforms. And so it's here, this is the data I showed you at the beginning that we had measured. That's a prediction. So that's what you predict using this model. I just put in the geometry that we have observed, and you can run a simulation. And what you get using this energy function that I showed you, you can get a profile. Well, the gradient is quite close, and even the anisotropy, which is integrated here over time, falls quite close to what we observe experimentally. Okay, so questions about that. So what I've tried to do is start with a complex process of tip growth. It involves many different things. Take it down a notch, if you will, in terms of complexity, describe the mapping of that, and which involves two functions. And I'm telling you here that, in fact, given the mechanics that we have understood about the cell wall, it seems as if we can predict how it expands just based on the geometry. So we can look at the cell, look at its geometry, and tell right away, oh, this should be the deformation field for that cell. Okay. Questions about this? Yeah.
Where is the mass coming? The increase of mass.
We'll discuss mass. But so in this model, I can step back. So the mass is coming from the cell, but I'm assuming the question is, where do you put the mass in the equations?
Yes.
Yeah.
So is it a membrane with a constant thickness, or the thickness of the membrane varies?
No, I'm just about to talk about the change in the thickness. The way we see it is, so the cell will secrete new mass, so these molecules here, pectin. As it interacts, it gets integrated. So the word intussusception that was used by Thomson was also used by many Germans to mean a cutting and pasting process, if you will. You have cell wall, you want to break bonds because it's elastically rigid, if you will. You break bonds, and as you open up, because you've broken a bond, new mass comes in that space. Okay?
Question?
Yeah.
Where does the new mass that goes to the inside of the cell, where does that come from? Does that come through the membrane from the outside, or is it connected to a long thing? Or where does it come from?
I have two slides, one slide that describes two processes. There's secretion, so
Yes.
that's at the membrane, you can bring vesicles that will add mass.
Yeah, but there must be some source of stuff from the outside.
No, it's the cell. It's all growth from within. The cell is producing the mass that it needs to build the wall. So there's water and nutrients that come from outside, but the wall is not built from the outside, it's built from the inside.
I understand it, but where does the stuff from the outside get to the inside to do the building?
Oh. But there's photosynthesis, but even the fungi, for example, will just, the osmosis.
Just osmosis?
Yeah,
That's it.
so most of the small molecules can cross the cell wall and the cell membrane, and they have transporters, so they can transport material from the outside, whatever they need. Whether they take nutrients from your skin when you have a fungal infection, or from a rotting apple or whatever, they take what they need from the environment and transport it inwards to then build their own cell wall.
And does that happen uniformly all over, or does it happen in some place which you're not talking about?
Secrets, I don't have any secrets. I did forget to, this is, let me step back because that's a good point. We'll start with this here. As far as we can tell, this map is continuous. So we've never had a case where we saw, for example, one little area that was producing surface, like a point source, if you will. Having said that, it's biology, and there are many cases where, always exceptions, so in bacterial cells that have a simpler cell wall, it's a single layer of molecules that are attached together that makes a cell wall. In that case, you can find zones where there's essentially a fracture, and you're adding one new molecule, or a row of two new molecules. So in that case, it's not a continuous map. You have to take into account the fact that you have a finite number of load-bearing elements, you're breaking one, and you're adding more mass. In these cells, the wall is thick enough, and I guess the scale is large enough that we don't, at least at that scale, so it's still at the level of micron or sub-micron, we don't see inhomogeneities. We just see a gradient, a uniform function.
Jacques, I was wondering if you had any information from cell wall biosynthesis mutants or, say, biochemical manipulations, calcium chelators, that inform your model?
I do. I'll answer partially this question in a few minutes. Yeah, can I? Yeah.
So is your system always local, or it could be non-local, like have an integration in time, like when you define the energy function?
So, being local, no, what do you mean?
I mean the energy function.
Yeah.
You have to integrate from t equals 0 to the time of interest, or it's just a local energy.
It's always.
Like I was worried about some of the non-conservative part.
No, so this, at every moment, this equation is not historical. So you always have an equilibrium of forces in the cell wall, because it's a quasi-static process, and therefore you can always define the measure of this at every given moment, this energy function.
I missed the point about mu and nu, the two parameters. Do you choose them to get the best fit?
Yeah, so mu has no effect. In fact, I won't do that, but if you set nu to 0, for example, this is just a scaling. Nu is just a scaling of these circles, so it doesn't change the angle.
So it's a one parameter.
It's a one parameter, yeah. So we played with that. It's actually, I put nu there because it has the role of a Poisson's ratio in linear elasticity. And it falls quite close to 0.5. It's not 0.5, it's a bit higher than that. But so it looks as if that's all you need to know to predict how this cell will deform.
Question. It looks like the resulting system is the system of ordinary differential equation, yes or not?
Well, if you're doing in space, it's a PD.
No. You have something W is a function of psi, yeah?
Yeah.
Psi dot is equal linear function of psi, yes?
Sorry. Are you referring? What is
Yeah.
psi? You new here? This?
Yes.
Well,
Therefore,
This is a matrix, right?
yeah,
A constant.
it is not a constant.
Yeah, they're constant. Well, they can vary spatially, But this is my strain, that's my stress, and that's a linear relation.
W is quadratic function of strains, yes? W.
Of stress or
Of
strain,
strains,
yeah.
of strains also.
Yeah, because
Therefore, your equation, resulting equation for evolution, is linear ODE, yes?
can we talk after?
You can get explicit solution finally. Let us look at the equation.
Well, we can look at that.
Which
But-
is expression for W, yes? Let us come back one slide.
So maybe you can solve
Yes,
that,
look at this equation.
after the talk. You know.
I-
Time is.
Yeah,
Time is
time.
short.
So yeah, I'm taking the gradient of that function, and that function is specified at every time point. I don't need to integrate in time to find what it is.
Linear
It's, well, that's the equation. So this is the equation, right? This is the,
It's already solved.
yeah. Okay. So going through your question, so we have this is the little model here. I'm telling you, let's just take the gradient of this strain energy function to find how the deformation, the fluid occurs on the surface. And it works well, so at least with the data we have. And so the question is, what sets this term now? So that's the part where the biology comes in. And there are two processes that could take place, and I'm depicting one here. So I'm going to come back to these later on, but the idea here is you have a growing tip, you might secrete, those are little vesicles that bring material to build the wall. And you could imagine the wall that's holding the pressure is a thin shell on the outside, and what the cell does is it secretes material that will evolve, right? You can use enzymes to prepare it until it gets integrated into the cell wall. Okay, so that's one process, it's called secretion. The other one, which I haven't depicted here, but also takes place in some systems, is you have enzymes in the membrane here that are actively synthesizing material and delivering that material in the wall. So for physical terms, it doesn't make a big difference to you. In biological terms, it's two different types of pathways, if you will, to build the cell wall. One is synthesis of the membrane, the other one is just secretion of material you've produced somewhere else in the cell, and you just bring it there. Okay? So would like to know which one of these is giving us this function here. And with colleagues formerly from Cambridge, now they've moved on, we've looked at that. So we work with Rafael Carasosadas and Juan Francisco Ibanez and Etienne Couturier, who's sitting back there, to look at how the spatial gradient, this chemical concentration, could be set up at the cell level. And we worked in fish and yeast. They have the advantage of having very good molecular tools. And so what you're looking at here, you don't see, that's a ghost, right? But there's a cell here, and that's the fluorescence of a given molecule. So that's crib marked with a green fluorescent protein. So that's one molecule that's localized at the tip. Here's another one, and so many different molecules. And we've color-coded them. So in blue here, those are molecules involved in the synthesis at the plasma membrane, and that's a glucan synthase. So that's a molecule that's involved in building the wall here. And you see it's polarized, it's polarized in its distribution. And then we've looked also at 4 different molecules involved in secretion. So Sept, for example, and XO70 are part of a complex involved in secreting material. And you see that all of these molecules are also localized at the tip. So we can, in fact, start looking. If I'm saying I just need some sort of concentration profile and I can predict how these cells are growing, well, we have now a selection of different types of molecules that might specify this concentration gradient. So we worked with them, and I'm going to summarize many years of hard work in one slide. This is what we've measured. So we've looked at hundreds of cells and measured the actual profile of these molecules at the very tip. And they're still classified. So these green ones are for secretion, the blue ones are for synthesis of the plasma membrane, and the black ones they're called polarity factors. They're more upstream, if you will. They're controlling the activity of other molecules. And we've actually used these profiles, these concentration profiles, as our function here to drive the growth. And this is what we found. So this is in fact the standard, if you will. That's what we observe. So we call it aerial here. The closest one we could find, the profile that looks the best, is called T1. So that if you look at the shape here, there you can predict quite well what we observed. But then there is many different molecules, or in fact these 4 different molecules that are involved in secretion that all come close. So you have a number of cells, some of them better than others, predicting fairly well what we observe in those simulations. So they're predicting quite well what we observe in living cells. And you can see the difference by this tree here. So there's a clustering of the tree, means they look the same in terms of their profile. And the branch length here is a measure of the fitness, if you will. So the shorter the branch you have, the closer you are to this prototype. And it turns out these molecules that are involved in the synthesis of glucan molecules at the tip, they do a poor job. They grow. If you use these profiles, which are quite large, quite wide, you grow very fat cell instead of observing these narrow cells. And some of the factors are in fact more localized, and you get thin cells instead of getting the cell of the right size. So there's more to do, but we concluded that secretion is probably, the process of secretion is probably the best molecular marker of what happens in this equation. So if I were to just choose one control at the cell level, I would say, well, I would put in here the concentration of some of these secretion markers as my profile, use my relaxation of strain energy, and I would predict the strain rates. And so this is what you see. For example, this is using secretion, so that's the typical. They both start with the same shape, which is the shape we observed. If you use the secretion markers, you can grow them nicely. If you use the glucan synthase markers, you get fatter cell. And if I were to run it longer, well, then we get fatter. Okay, I'm gonna run out of time. Let me just mention. I can take 5 more minutes. Yeah. We did, so the slide I just jumped is a control we did to show that indeed secretion is controlling what we see there. I'll come back to it if you have questions. I just wanted to switch gear and show you some nice things these cells do. They do this. So they stall, and then they pulse, and then they stall, and they pulse, and then look, when they stall, the wall gets thicker. So it's stalling, and you see, this is the wall here that you see thickening, and then it pulses again, and so on. And we have some instability there. We're tracking, it's the same cell, but we're tracking the outline of the cell and plotting its pole velocity as a function of that.
What are you plotting there?
This is the velocity of the tip. So it's going slowly, and then it pulses. So it's quite a remarkable oscillatory process.
Is the motion inside of cytoplasmic streaming that brings stuff to the cell?
It does, but it's actually this streaming that you saw in the cell, which is quite striking, is goes beyond, if you will. You don't see the actual vesicle. This is starch, so it's really food stuff. The vesicles are down here. You don't see them. So the actual secretion process is affected by vesicles that are at the very tip.
But the rotation brings the vesicles over there, or what?
Some of it, but most of it is actually moved along actin filaments. So it's targeted. There's polarity in the actin filament. It's targeted at the very tip. Okay, so we'd like to explain that. And one feature that we've found quite striking is in the cells that pulse very strongly. This is petunia, yes. This is pollen 2 also of petunia, that almost looks dead, looks like it's not doing anything, and then it will move forward quite quickly within 2 or 3 seconds, and then it drops dead again for another period of time. And if you look at the velocity as a function of time, you see this. So we're recording noise, if you will, and then at one point, it speeds up and dies nearly as fast. And you see that again and again. Now, plotted as a function of time, what you see is the period is actually not very constant. The spacing here, so the period of time between different pulses of growth can change and can be quite long compared to other ones that are quite short. If, instead of doing that, I plot in terms of the distance, right? And in fact, you see it here. Look at the spacing between, this is distance, right? The spacing between the pulses, it's perfectly regular. And here's, I've got it, so this is distance covered by the tip, and the same function here that we're plotting, the velocity, but along this axis, the distance, the spacing, wavelength now, is perfectly constant. Okay. So why? Why is, when you think of oscillating system, very often when you think of a pendulum, and the pendulum is very good at keeping the period. You can, in fact, push it a bit harder, and it's good for in a clock because it wants to keep a constant period. These cells are not like that. It's as if they want to keep a constant step, but they can sit there and think longer or shorter, and then we'll take another constant step. So why do they do that? And if you're not convinced, well, we have period 2 orbits, so cells that will have double peaks. They get excited, and then the peak will peak twice. So here's 2 oscillations, and then a lull, a period where it's not doing very much, 2 oscillations, and so on. And if you, this is a chymograph. If you're a biologist, you know about chymographs. If not, you probably don't know about chymographs, but it doesn't matter. I have space here and time, and what I've marked, these points here are places where the cell was peaking. So if I project in space, you have a perfect spacing, perfect wavelength. If I project in time, I have the 2 periods that you see there. So the long period that corresponds to these here, a short period that corresponds to these over here. Okay? So even a cell that's obviously within a period 2 orbit will decide to take steps that are always the same. Not perfect, you'll see there is noise, but so we're trying to understand why that is. So we started making a more detailed study of the kinematics of a cell that's oscillating. And what I'm plotting here is 3 different measurements that we make, or 3 measurements. So the period here along this axis, wavelength, and then velocity. And velocity is in fact defined based on the wavelength and the period. So velocity is just wavelength divided by period. And those are all cells, and the blue points are all different cells that we've measured. And red is one cell that we've tracked over a long time to see if a single cell will visit essentially the same space of parameters, whether it's ergodic or not. And it's somewhat ergodic. This cell that in red that we tracked over a long period of time managed to cover quite a bit of the typical range that we see for these different cells. Now, if you look at let's look at wavelength and period. The period goes from over 100 down to about 10 seconds. So the shortest period have about 10 seconds. The longest one are over 100 seconds. So we have at least a 10-fold difference in this parameter. If you look at wavelength, it's not constant, as I might have indicated before, but it's more narrow. So you go between 2 and 3, something like that is the minimum value, and the maximum value you see is something like 10, and therefore you have a factor of 5, perhaps, of this parameter. And so we wanted to explain it, and the surprising thing for in our thinking, if you will, is how, if you have a cell that pulses like this, and the velocity seems to be not doing anything for a long time, what is evolving in the cell if it's not the velocity? There must be a variable that's evolving, and at some point goes over a threshold and makes it a spike. And the velocity obviously is not that, because the velocity seems to be flat. And so we've been looking for what is this covariable, if you will, that is keeping track of time or space while the cell is not advancing or moving very much. And cell wall thickness is secretion of wall material so that you can grow, has been the recent conclusion. And I won't be able to go through these details. This is a paper from the Hepler lab, where they showed, to just give you the central message, they showed that the wall thickness, or the thickening of the cell wall, precedes the growth by about a quarter phase. Okay, so you have, as the cell gets thicker, its wall gets thicker, it gets ready to speed up. So the peaks of wall thickness are just preceding by about 90 degrees or a quarter phase the maximum in growth. And there is an old theory in biology called morphogenesis, if you will, called stored growth, and it's an idea that people have seen in plants where you can inhibit growth for some time, and when you relax that inhibition, very often a plant or a cell will speed up to recover the time it lost waiting. So it's as if they wanted to grow, they probably had secreted the chemicals they needed to grow, but perhaps you remove the pressure, and therefore they didn't have the force there to deform the surface. And as soon as you recover the pressure, they start accelerating, and they recover whatever time or distance they might have lost in growth. And so we wrote a little model to describe that. It's a non-spatial model. I'm just looking at the pole here, how it's moving. And I have two variables to model this. And so I have velocity, which is just the speed of the tip here. And I'm going to measure, so I'm going to assume that the h here is the thickness of the wall that's holding the pressure, and rho is the thickness of the stored growth. So the material that will hopefully at some point be integrated in the wall and allow the cell to grow forward. And so rho will evolve in time, so as will the velocity. Rho, the equation we wrote, is just a conservation of mass. So this thickness here will increase when you secrete. So that this is a length, so that's the velocity. This is a velocity as well. This is the rate, the flux, if you will, of material across the membrane. And then it will decrease by mass conservation is when the cell moves forward, its material is advected and it exits the tip, if you will, at the equator with a thickness h. So that's the, if you will, the loss of mass because of advection of that material. The other equation that we have is for the relative velocity, and I probably go over that later. But we just said that the relative velocity will be, or if you will, the acceleration, or dv/dt, the acceleration will be positive when the stored growth is above a steady state or critical value, and it will be negative when the stored growth, so this thickness here, is below this critical value. So very simple equations with only two parameters. This h and r are fixed, it's just the geometry of the cell. S is one parameter and alpha is one parameter. And this is, actually this equation don't, at least as far as I know, don't have an explicit solution, but you can write them as a potential. So this is, if you will, the potential that describes the path that have to follow these equations when you solve them. And for a given set of parameters, this is what the surface looks like. If you pick one of those, there are many paths that are all parallel, so you don't converge to a limit cycle. There are many different cycles that coexist. If you're on a cycle that's close to the steady state value, you see this is the growth, the velocity here, so that's, you see a nice sine wave velocity, and the axes are different here, but this is the stored growth in red. If you happen to be on an outer curve here, this is what you'll see. So you'll see long, for the growth, for the velocity here, you have long period of no velocity, and then very sharp peaks. And during the time where you have no velocity, the stored growth builds up until it reaches a critical value, and then it crashes as the cell accelerates. And you have essentially the right phase relationship, given that we have almost no parameters. So you peak, you have a thicker or stored growth, if you will, before you start to see the cell accelerating. So we use this model, and we show that we can actually fit with these two parameters, we can fit the data. This is the fit of the data for the velocity versus period. This is wavelength versus period, and so on. So I'm over time, so I'll just show you a movie, and you ask me a question. I'll show you like two movies. This one is it's called Stuck in a Corner, and you see what you do when you're stuck in a corner. This is a pollen tube growing through a maze of obstacles. So those are little corners made of agar. So if you're strong-minded and you have enough pressure, you can push your way through agar and bend over. Or you can, if you're really cornered, you can bend over backwards. There we go. So we're trying to understand how these cells grow in complex environments. They have to negotiate other cells, and fungi, for example, that do invade your skin, they have to find their way in, and so on. So we're trying to understand how they mechanically manage to penetrate a stiff environment. Start with that.
I'm a little confused, so can I see if I understand correctly? You showed a model which, as if I understood correctly, had two heuristic sort of insights. One regarding this function W, dependence on strains, and another basically chemical potential or whatever you call it. In this model, does it? You show that it predicts under some circumstances with some parameters the shape evolution, but does it predict the speed with which these dendrites grow correctly?
No, the speed is difficult to predict because even nature doesn't have a fixed mind about the speed. You can grow cells that are all coming from the same organism, same culture. You grow them side by side, and one will be growing fast, and the other one will be growing slow, and you can't tell why.
That's why you're developing the second model.
But even that, so this second model explains oscillations. If you're to ask me, predict for all of these cells what are their typical characteristic velocity.
So how do I put these two things together? That I have some understanding of the evolution of shape and time.
The two models you mean? Or?
Yeah.
No, the oscillation and the oscillation that I showed you is a growth process. We understand. I cannot tell you what will be the mean velocity, but this simple model, which we'll do in dimensional, it's now just non-dimensional. But if you do it dimensional, it would be if you will, the time varying solution of what I showed you in the first half of the lecture. So this growth pattern that this constitutive equation, really that's what it is, that predicts how the cell surface is deforming. If you add to that a reservoir or stored growth, you can get instability, you get cells that oscillate. So you could predict both the profile of expansion, but also why in some cases it oscillates in time. Predicting the velocity, which would be something most physicists would like to do, can't do it. There is no, it's biology. We can make it slower. We can make it grow slowly by giving it poor conditions to grow. But in terms of telling you why these cells grow faster on average than others, few people know, but we don't know.
There was one question.
So what is the trigger for this release of flow?
The oscillations.
So when it reaches some threshold, what's the nature of trigger? Why?
Yeah, so this is where we'll need a spatial model. What I think is going on. So there is built. So we're showing with this model that when you reach a critical thickness here, then it starts growing. I think what it means it means that the material, if you have a thick layer here, it means some material was secreted a long time ago, right? And if it's secreted a long time ago, it means it's right against the wall here. This is the wall that you need to deform, and it has the molecules that are primed to be inserted. Because the problem we have in this system is what you secrete, I put it in yellow, because it's not ready to be integrated. It's a molecule that's methylated, and so cannot be integrated. So it needs a transit time in here to arrive, to be primed and be integrated. If it's thick, it means it took a while to get there, or the molecules that are here have been there for a while, and therefore you can expect fast growth. That's in a nutshell.
Is there a follow-up question to that same one? I mean, you've represented the wall for simplicity's sake as a homogeneous domain, and of course it's not, and you've got cellulose and pectin layers and other interactions going on there.
In the cells, no. So in pollen tubes, it's just pectin.
In plants, I think, sorry. But I'm thinking there are mutants in plants that cause disruption. And I'm just wondering, because there is an argument that you've got two processes that need to be balanced. There's the maintenance of integrity and the need for plasticity or viscosity. And I guess the question, if you look back to the way the primary wall is put together in normal cell divisions, you've actually got different carbohydrates involved, like callose-based systems, which provide that first template for secondary strengthening. So you've got this issue of formation and then strengthening. And I'm just wondering, is there an intrinsic reason why you got this pulsatile nature, that you got two processes in your analogy of walking, the stepwise forward, where you've got two alternate processes that need to be maintained in obviously balance when you're walking? But is that another possibility?
Well, for the lily pollen tubes, it's only pectins. So there is no, when they do microscopy on the tip, there is no cellulose. There'll be cellulose later on, there'll be
I
callose.
wasn't actually thinking about the two layers, but the two processes, say rigidification versus plasticity. I guess that would be part of your model here.
So that's what we don't spell out because I don't want any parameters, free parameters, but that's what will be going on. Yes, secretion is not enough, so you can secrete material, but until the enzyme is called pectin methyl esterase, until that enzyme has acted on the material and prepared it to be integrated, it serves, right? This material is useless. It's just mass, dead weight, if you will. It cannot be integrated. It doesn't have the reactive groups to be integrated in the wall. So there are certainly processes, and I think that's an important part here, of preparing, if you will, chemically altering these molecules so that they can be integrated in the wall.
I'm just trying to see why you couldn't have a continuous way of managing that. That's your prediction, isn't it?
No, you see the cell oscillating. I don't want to predict it.
The standard growth mode is constant velocity, so...
So the pollen tubes are in fact quite unstable. After they've grown some time, most of them will oscillate. In fact, we had a hard time finding cells that don't oscillate. Well, hard time. Say 10% of the cells will grow steady. So they, at least in culture conditions, the pollen tubes have this tendency of becoming unstable, so they will oscillate.
Is it related?
Related to this, maybe a way of rephrasing this question is, what sets the period of oscillation? You have a whole bunch of parameters in this model.
No, not a whole bunch. Careful. There are two.
All right, well, more than one.
Yeah.
Yeah. So, what sets how does the period depend on these parameters?
The interesting question is why the wavelength is more constant than the period. So that's really what they're related, huh? But so that's the intriguing part to me. Why is it that these cells care more about wavelength than about period? Right? It's not.
But the previous question, if I correctly understood it, was about why you have oscillatory behavior, and I'm asking a follow-up question.
Yeah. So,
what sets the period of oscillation?
So in the model, it's this, right? It's these. Well, it cannot be anything else, right? Where am I? So it's the S is the rate of secretion. This is, if you will, the chemical reaction, because that's the growth, that stored material, and that's the velocity which comes from integrating new material. So this is like a rate. Sorry, this is like a rate constant. And so in the model, all of these curves, right? They're set, or if you will, the surface of this profile is set by these two parameters. So if you change these two parameters, you'll get different periods, although it doesn't change very much. So this is if you were in Chile, that would be an earthquake, but we're not, so it's just a truck, I guess. And mostly in Chile, people run out as soon as there are some vibrations. So in this, actually, if you look at the space, the period here, it's quite similar. And so we've got very short cycles here, very big ones. So in this model, you get a fairly fixed, for a given secretion and alpha, you get a fairly fixed period. I don't have the exact. I have written somewhere the equation, but it's not analytical because we don't have an analytical result, so it's just inferred from many simulations. It was. Oh, it's written.
You can go and see him
I'll check
later or tomorrow. Julius, you had one question?
I was going to ask, in this, you have this model where if you plugged in the wrong CFS, the wrong chemical species, they started growing.
They get fatter, yeah?
They get fatter. That surely reaches a steady state, or does it just grow? Does that
No, they
grow?
do reach a steady state. They, in fact, and what I showed you.
So isn't it just a rescaling in some, like if you had started with that thickness, you would see normal growth, wouldn't you?
Yeah, but what we're trying to do is this is it, right?
Yes.
We're trying to see what profile maintains the cell we observed. So what profile is compatible with what the cell is doing?
And you're saying you couldn't have changed some of the other parameters to get that CFS?
No, but you can. What we've done, so you're right. I'm assuming that it's there. This is a bit dizzy. I'm assuming that there's a very simple relation between this profile and this, and it's not like that. First of all, this profile is a measure of fluorescence. Has nothing to do with concentration. In fact, it can be correct in terms of concentration. Doesn't mean that it's active, the molecule, but this reflects the activity of these molecules. And it could be nonlinear. You could say this profile to the x power gives me this concentration. So I didn't put it there. We actually played with the parameters to show how they affect the geometry. And yes, I could get all of these to fit perfectly by adding more to it, by either changing the exponent or making some nonlinear assumption. And that's why I had this slide here, sorry. To confirm, so Sib here is a molecule that's involved in secretion. You can delocalize it. This is a cell, this is the normal cell here, which Sib is here. In this construct here, you have Sib over a very large area, and the cells are fat. So we've played with this one to show, look, if I change now the secretion, I make it fatter, wider, the distribution. Do I see that in the cell? And in this case, we see it.
Okay, so, well, Jacques is here and still today and tomorrow, and you're leaving on Monday, right?
Yeah.
Okay, so let's thank him again. And we might go and ask him other questions.
One second for now.
That was the plan. He left. So
Wait.
let's see.
Wait,
Yeah.